Hello, hello, hello. How's everybody doing? Good to see you folks. Everybody doing well? Can you hear me right now? If you can, go ahead and give me a check in and let me know that you can hear me. Uh-oh, yep, there we go. Fantastic. Sounds like I'm in. Good to see everybody. Wow, we have got a good crowd. Ashton, Camilla, Stephanie, Etta, Michaela, Tara, Camilla, Kate, good to see you. I got Lily on my remote phone, so it's good to see you. Harrison, Tyler. Wow, what a class today. Fantastic. I'm glad everybody took the time to come today. I do appreciate that. It's good to see you as always. We got 10:59, so we'll wait just another second and then we'll go ahead and get started. Fantastic. It is so good to see everybody today. Coming to you live from Wake Tech's South Campus, the Social Science Resource Center. I am sorry about the technical difficulties we had last week. We had two lectures and one of them went off well and one of them didn't. So now, batting 500, I'm a baseball guy, so that should put me in the Hall of Fame, but I want to bat 1,000 for you folks. So I went ahead and bought new equipment and moved my entire studio. So hopefully things will go a little bit more smoothly today. Emma, Jordan, Tatiana, Alma, wow, good to see everybody. Fantastic. Hi, Laura. All right, so is my sound okay? Too loud? Are you hearing popping noises, or does it sound good? Sorry, I can't quite see with my glasses on these days. Holy cow, he's getting old. All right, there we go. Fantastic. All right. Sounds like we've got a good start. Well, you know what? It is 11 o'clock, so let's go ahead and get started. I'd like to welcome you to week two. So last week was kind of a free-for-all, everybody trying to figure out how the course worked and get your homework and your discussion board turned in on time and figure out the pace of this class. So today is a big day to sort of recalibrate ourselves and get off on the good foot for the rest of the semester now that we know how this course works. We had our first weekly competition with the Neuromatrix last week, and it was totally exciting, so I'd like to go ahead and send you to my colleague over at the Neuromatrix Sports Desk. Well, thanks, Chris. We had a great weekend of competition in the Neuromatrix with 102 people qualifying to participate this week. Wow, what a big competition. You see, we've got the top 20 here in front of you, and I'd like to give a special shout out to Weewa, La Squishy, and Kamel for finishing 1, 2, 3. And you'll see that the top 20 is rounded out by McConroy, Kircho, and Che. Fantastic. Congratulations to all the competitors this week. The Week 2 Neuromatrix, Calling Dr. Pepper, will be available on Friday morning at 9 a.m. This Neuromatrix contest is going to cover all the parts of the brain and will be featured prominently in the Thursday lecture, so you do not want to miss the lecture on Thursday. I had a couple of people say that they needed a little more time to answer the questions, so I've added 10 more seconds to each of the Neuromatrix questions from here going out. Hopefully that'll give you enough time to complete these questions. And you know what? You may not have finished in the top 20, but that's okay. You get 1 point of extra credit on your collaboration grade each week you qualify and participate in the Neuromatrix contest. Remember, in order to qualify, you have to register and do that week's homework. Finally, we've still got 14 more weeks of competition, so there's plenty of time for you to work yourself up into the top 20. Chris, back to you. Wow. How about that competition? Congratulations to Weewa. Very impressive performance this weekend. Great job, and I hope to see everybody participating again this week. Now, the second thing is we had our first discussion board this week, and instead of providing feedback to each and every person, what I'd like to do is just provide a general bit of feedback to everybody in the class every week. And so we're going to have a feature in this class called the Opinion Desk that we're going to run every Tuesday. So without further ado, I'd like to send you to Chris over at the Opinion Desk. Thank you, Chris. Welcome to the Opinion Desk, where I pose the questions every week and you make the decisions. This week's discussion regarded the subfields of psychology. I provided you a very interesting TED Talk by a fellow named Deb Roy, who documented the emergence of the word water in his young son. And what I was interested in finding out was what area of specialization, what subfield you thought would be most interested in this psychological talk. And you folks came up with lots of answers, and most of them were correct. So what I'd like to do is go through and talk about the answers that you came up with. Now, of course, everybody thought developmental psychologists would have been interested, and you're right, because they would be interested in how a new language emerges in infancy. Good choice. Some of you also thought that a cognitive psychologist would be interested because language is a cognitive ability. You also would be correct. Some of you said social or cultural psychologists, and you would also be correct because they would be interested in the effect that our social environment has on the development of new behaviors. Now, the social psychologist would be interested in the talk from the perspective of how groups generally function, and the cultural psychologist would be interested in how different groups function, but they would both be interested in the social environment. Some of you said educational psychologist, and you know what? You're right, because the educational psychologist would be interested in gleaning insights into how language is learned. Any of you taken Duolingo before? It uses the same approach that Deb Roy seemed to document. And finally, a biological psychologist probably would have wanted to throw an EEG or a PET scan or an fMRI in there and try to figure out what parts of the brain were active when the child was learning the language. Very good choices too. Now, I didn't see this, but I think an industrial organizational psychologist would also have been interested in the last 5 or so minutes of the talk, where they were talking about using big data to understand how social influencers in the media worked. So I think an IO psychologist would also have been interested in this talk. So almost all of the areas of specialization would have shown some interest in this particular talk. Now, okay there, folks, how you doing? Looks like that didn't work out too well, so we're going to go ahead and can that. Is everybody doing all right? Uh-oh, now I've gotten choppy. Fantastic. I realized what I did. All right. If you could, give me 1 second. Okay, we should be working now. I won't do that again. Okay, so it should be back. Okay, fantastic. You know what? I won't do that anymore. I thought I could play video clips in here, but apparently I can't do that, especially for clips longer than about 1 minute. Okay, fantastic. One thing I did want to do before we go on to the lecture is answer one quick question that I got from some students, and I know I can do this without messing up the buffer. Okay, so let's go ahead. Some of you asked, why is my final collaboration grade 104 out of 132? And I wanted to take a minute to explain exactly how that was going to work, okay? So your collaboration grade starts out as 100, remember? Okay, so you've got a collaboration of 100. Now, I told you that I'm going to give you an opportunity to earn some extra credit in 3 different ways. And so these are the 3 ways in which you're going to earn extra credit in this class. I told you that Remind was very important to me. So if you signed up for that, I gave you 2 points extra credit on your collaboration grade. Now, if you do come to the webinar and do the discussion board every week, you'll get 15 points of extra credit, 1 point each week times 15 weeks. And if you participate in the Neuromatrix contest every week, you'll also get 1 point times 15, so 15 points there. So if you add all of those up, your total possible score is 132. Now, because we've done 1 collaboration, 1 Neuromatrix, and you've signed up for the Remind, your best possible grade right now is a 104. So hopefully that helps you understand where your grade is. Does everybody understand that? Does that make sense? If you would, please go ahead and type in the chat bar and let me know. Okay, so let's go ahead and get started with the lecture if we could, okay? So it can still go up. Yes, it can, Aaron. In fact, if you do everything, all the extra credit activities at the end of the semester, it can go up to 132 points, right? But you have to do a lot of extra work in this class to get that. Now, before we go to the lecture today, I just wanted to point out to you some resources that you're going to find in the course resources folder of this class. As you just saw with the frozen video, I can't use very long video clips in this live stream production studio. It messes up the program. So I'm going to be describing things today that are dynamic processes, how a neuron works, how information moves along it, and these things aren't really easy to explain in a PowerPoint presentation. So I've got some video animations that you should watch, and they're going to explain the processes that I'm talking about today. So hopefully that'll give you a little bit of insight into what we're talking. If you have trouble after the lecture, please feel free to go to these videos. Okay? Now, Chapter 2. Chapter 2 is all about biopsychology. Biopsychologists are interested in how the structure and function of the brain is related to our behavior, our thinking, and our personality. Good to see you, Ross. Fantastic coming into class. We got a bunch of people here today. And so today we're going to talk about it from a cellular level. We're going to talk about the basic building block of the nervous system. We're going to talk about neurons and how they communicate using these chemicals called neurotransmitters, okay? So what we're going to talk about today is the organization of the nervous system, a little bit about the anatomy and physiology of the neuron. We're going to talk about the function of neurons, and then we're going to talk about hormones as one form of neural communication. All right, so let's go ahead and move on. So neurons are the building block of your nervous system. They suggest, I don't know who counted them, but they suggest that there are 85 to 100 million of these information communicating cells in our nervous system. Their job is to allow one part of the body to communicate to another part of the body. They're communication cells. Turns out that we have all the neural pathways that we'll ever need present at birth. And these neurons come in all different shapes and sizes. Now, along with neurons, your nervous system is made up of these things called glial cells. They're support structures for neurons. I was just looking at the question over here. Haley had one, but it's not a question I need to answer. So the glial cells are support structures for neurons. Now, some of them speed up the neural impulse and help the neurons communicate faster, and some of them clean up intracellular waste. And it turns out there are 10 times as many glial cells as neurons. And these are the part of your nervous system that develop after you're born. And we don't have these glial cells at birth, and this is why infants are so helpless at birth. And we'll talk about that more in Chapter 4. Hi, Highness, it's good to see you. So what you see here are 4 different kinds of neurons and 2 glial cells. The astrocyte is a glial cell that's useful in helping neurons communicate, and the microglia is a glial cell that is responsible for cleaning up intracellular waste primarily. Okay, so let's talk about the organization of the nervous system. How are we going to organize these 100 billion neurons in our brain, in our nervous system? We're going to talk about them as sort of existing in 2 systems. The brain and the spinal column are the parts of your nervous system that receive and respond to information. They are the decision-making control centers of your nervous system. All right, your brain controls your muscles, your internal organs, and your higher thinking. and your spinal column is responsible for reflex responses. And so we're going to differentiate the central nervous system from all the other neurons in your nervous system. And these we'll call peripheral nerves because they are outside of the central nervous system, the peripheral nervous system. And these are the neurons that connect your central nervous system to the rest of your body. Now, if you think about your body, let's break that down into two systems. There's a system that controls your muscles, your musculature, and we'll call that your somatic nervous system, and those are called somatic nerves. And we'll contrast that with your internal organs and all the neurons that control those internal organs, and we'll call that the autonomic nervous system. And you can think of autonomic as automatic. You don't control your organs, but you do control your somatic nervous system. So this is sort of a good way to think about think about the nervous system. So, you know, actually, when we use the term nerves, some people use the term nerves and neurons interchangeably. They're actually not. A nerve is a bundle of neurons. So all those blue things that you saw back here, these are all nerves. And a nerve is a bundle of neurons. And you'll see two pictures here. On the top is a cartoon picture, and the picture below is an actual nerve that is stained. And so you can think of nerves or neurons as being of two types. You sort of have these motor nerves, which carry information from your central nervous system to your brain. So I have a motor nerve that moves my hand around. And then we have neurons are one-way communicators. So if you have a motor nerve going out, you need some information coming back from that finger. And that's called a sensory nerve, which is composed of bundles of sensory neurons. All right? And for an example of a sensory neuron is a sciatic nerve. Anybody have a grandparent or ever experienced sciatic pain before? And that's just a nerve coming up your spinal column that gets impinged, and that's what causes that pain. Now, each of these axons becomes wrapped in a layer of fatty insulation after birth. This fatty insulation is composed of glial cells. Exactly, Ash Guth, it is sciatica. Fantastic. Okay, good deal. Hyatt goes by Jesse. I will try to remember that. Oh, no, Lorena actually has sciatica. That's kind of a bummer. I have had some spine and some pain right there at the top of my hip before, spinal pain, and it is definitely not a very comfortable situation at all. So each of these axons, so actually these nerves are not really composed necessarily of neurons, but a part of a neuron called an axon. Think of the axon as the wire that connects one part of your body to the other. And if you look at the top picture here, that little yellow string coming out of the picture is an axon. And you'll notice how it's wrapped in a layer of what looks like cellophane. Those layers are actually myelin sheath. And myelin sheath is just layer after layer of glial cells. Remember I told you glial cells? So glial cells wrap around your axons and help them transmit information more quickly. Now, although neurons do come in many shapes and sizes, there's one common anatomy diagram of a neuron that most introductory students, psychology students see. And you are looking at it right here. And so what I'm going to do is I'm going to go through and point out some of the important anatomical parts of a neuron so you can understand the mechanism for how it works. Now remember, this is the part where I'm talking about actions that are happening, dynamic actions. So as I describe them, they may not make perfect sense to you, but if you go to the videos that I have in the resource folder, they will really complement what I'm saying very well. So you should definitely do that, all right? Now, so neurons are one-way communicators. That is, over here, right here at one end, the dendrite and the cell body, these structures collect information in the form of chemicals, and it causes the neuron to either be excited or inhibited. And if that neuron is excited, it sends a message that goes all the way down. Let me see. Whoa, down the length of this neuron. Like this. Okay? And so neurons move from this direction to this direction. So I'm going to start describing over here, and then I'm going to move myself back as I talk. Okay? So the dendrites over here, you see all these little tree branch-like structures over here? These are dendrites. And what they are is they are these little root-like structures that contain protein receptors that have sort of a lock-like structure. And they are looking for these chemical molecule keys to fit in them. So your dendrite is this root-like structure, and it's going to have thousands and thousands of these receptor sites that are sensitive to one or more certain types of chemical structures. It's like an information gatherer, if you will. And so what they do is they receive chemical messages every second. Your brain is excreting these little chemicals into these around these dendrites, and these chemicals may tell that dendrite to become excited. They may be excitatory chemicals, or they may tell that dendrite to calm down. They may be inhibitory chemicals. I was just looking over here at the questions for a second. All right, and so you've got these excitatory and inhibitory messages coming into the dendrites, and they're gathering this on their receptor sites. Now, what happens is this information is then transmitted to the cell body, which is this next structure. It's that cellular structure up right there. And you notice how the dendrites are connected to that cell body structure. Now, the cell body contains the cell nucleus. Just like every other cell in your body, a neuron has a nucleus with ribosomes and DNA, et cetera, et cetera. But this cell body also takes this stimulation, and it has this little spot right at the very root-like structure called the axon hillock. And it receives these excitatory and inhibitory messages. And if it gets enough excitatory messages, what it will do is it will become depolarized, and it will send a brief pulse of electricity down the length of this axon. Okay, it'll go all the way down the length of this axon. And so think of the cell body as the decision maker of whether or not this cell is going to communicate to another part of your body. Have I lost anybody yet? It's hard to see your faces, so I don't know if anybody's lost. If you have any questions, please feel free to type them in the chat bar. Now, you have this long root-like structure, and that's how your neuron communicates from one place to another. So it is stimulated over here at the dendrite, right? And if it decides, it sends this brief pulse of electricity that travels all the way down to the end of the axon. Now, some axons are microscopic in length, but some axons actually can be as long as 3 feet long. Your sciatic nerve has axons that are almost a meter long that communicate from your lower limbs all the way up to your brainstem. So think of one neuron, one brain cell that runs almost 3 feet. And what happens is this brief pulse of electricity travels all the way down the length of this axon till it gets all the way down to these things called the terminal buttons. And we're going to go to the terminal buttons. I'm going to skip over myelin sheath for now. Now, the terminal buttons are where you have collections of chemicals called neurotransmitters. Remember I told you that neurons are sensitive to neurotransmitters? Well, you know what? Down at the other end of the neuron, they have neurotransmitters that they release to try and activate other cells, and that's how they communicate one to another. So down at the, down here at these terminal buttons, you're going to have collections of neurotransmitters. And when they're stimulated by electricity, an action potential is a depolarization that results in a brief pulse of electricity. When they're activated by electricity, they dump their chemicals out so that they can stimulate another dendrite. And that's how a neuron sends a message. It receives a message over here, and it travels all the way down and sends another message to the next neuron. And it travels all the way down the length of this axon. Now I'd like to step back for a minute and talk about this myelin sheath. I showed you the picture of the nerve a couple of slides ago, and I told you that the axon was wrapped in this insulative tissue called myelin sheath. And so myelin sheath is a fatty coating on the axons of these larger neurons, and it insulates the axon, and it actually speeds up the action potential and makes your neurons work more quickly and more effectively. And in fact, one of the things that allows children to start developing motor control, language abilities, and their intellectual functioning is the growth of this myelin tissue along axons that work these particular abilities. And as these axons become myelinated, the child's behavior matures. And I don't know if any of you have heard of Lou Gehrig's disease, amyolateral sclerosis. That's a demyelination disorder. What happens is this myelin tissue starts to die off around the axons of these larger motor nerves, and it causes you to become debilitated and eventually to die. And so myelin is a very important part of the nervous system. All right, so now what I've got right here is a blown up picture of a terminal button. Do you see this button looking structure? Let's see, this button looking structure right here. All right, this is a terminal button, and it relates to this picture over here. So you see the dendrite in the terminal button in large scale. It blows it up so that you see just one little terminal button. And this is a protein receptor site on a dendrite. This is postsynaptic. And then this is a presynaptic terminal button. Now, in the presynaptic terminal button, you have these molecules called neurotransmitters. And we're going to talk about them in just a minute. And these molecules are waiting in this terminal button. And when they're stimulated by electricity, what they do is they diffuse through the wall of the terminal button, and they float into this space right here between two neurons. Neurons don't actually touch. They have a little bitty space in between them. And what happens is these chemicals diffuse from the terminal button. They float over to the receptor side, to the postsynaptic side, and they lock into those receptor sites because they're shaped perfectly for those receptor sites. And they may either send an excitatory message. They may either tell that neuron, the next neuron, to be excited and send an action potential, or they may be inhibitory and tell that neuron, hey, whatever you do, don't send an action potential. And you've got thousands of these little terminal buttons, and they're each releasing a few molecules every couple of seconds, depending upon whether or not they're stimulated by electricity. And as soon as these molecules send their message, they are sucked right back up into the terminal buttons. Most of them are, but some of them aren't. And the remaining molecules, as I say up here, are metabolized. And they're metabolized by glial cells that eat up waste molecules in your synapse sites. And so that's the other function that your glial cells work. They work right here in these synapses, and their job is to eat up extra neurotransmitters. Okay, so the axon is kind of like a shipping department. The package comes through the dendrite and then travels down the axite to the terminal buttons. Riley, see, that is a great way to think about it. And the myelin sheath is kind of like the cops protecting the package. Let's think about it this way. The myelin sheath is sort of like the jet plane that speeds that message along, right? So think of it actually, yeah, I would think of the the cell body as the shipping department, and the axon is the super fast highway that the truck ships right down. That's a great way to think about it. I like your analogy, O'Reilly C. Thanks for helping out there. That's great. So this is sort of what happens with a synapses. Now, your book talks about these chemicals called neurotransmitters that are the chemicals that work in your nervous system. And so these are molecules, teeny tiny molecules that your brain makes and then secretes. Now, you have almost 100 different chemicals that work into your brain to regulate your functioning. However, the 7 neurotransmitters that I have here on this chart pretty much do about 95% of the work. So really, if you want to know what neurotransmitter function is about, really you need to learn these 7 neurotransmitters. On the exam, I reserve the right to ask you questions about these 7 neurotransmitters, okay? Now remember, some neurotransmitters are primarily excitatory. When they're released, they cause the next neuron to become excited and want to send an action potential. Some neurotransmitters are primarily inhibitory. When they release, they depress. When they're released, they depress the activity of the next neuron. But you know what? Most neurotransmitters can be excitatory or inhibitory depending upon where they are released in the nervous system. All right? So briefly, I'm going to go over these neurotransmitters. Acetylcholine is very important for attention, memory, learning, and sleeping. Okay, think about it. On the test, I would ask you about attention, memory, and learning. It's definitely a focusing neurotransmitter. Norepinephrine is that neurotransmitter that is released in your heart when you see a bear that scares you, right? If you've ever been scared by a bear and had an autonomic nervous response, your brain has released norepinephrine. Your peripheral nervous system has released norepinephrine. Serotonin is a mood regulating neurotransmitter. People who have been diagnosed with depression sometimes take drugs that elevate the level of serotonin in their brain. Then there's dopamine. Dopamine is important for reward and motivation, and it is implicated in addictive behaviors. Does something big trigger reuptake versus molecules being? That's a good question. You have the transporters that go from the presynaptic site into the synapse. There are a certain number of them, and they release a certain number of chemicals, and then at the same time they work in reverse. So basically, the reuptake happens at the same rate to a certain degree. It's based on the number of transporters that you have in the presynaptic site. And then you just have these astrocytes that clean up what is left over. So I guess because there's molecules are going in and out the same door, it's sort of regulated by the number of presynaptic transporters. It's sort of a negative feedback loop. As soon as they're released, the transporters start working in reverse. You know what I mean? So how does norepinephrine relate to adrenaline? They're essentially, norepinephrine is an epinephrine, noradrenaline is adrenaline. They're sort of the same thing. So noradrenaline and adrenaline are both in the peripheral nervous system, and I'm not a biological psychologist, but I believe in the peripheral nervous system, these neurotransmitters are excitatory. They cause your heart to beat faster. But in the central nervous system, in the frontal lobe, they actually work in an inhibitory function and are implicated in ADHD. So we are thinking alike. Oh, fantastic. I'm glad everybody's thinking alike. Okay, GABA. If you've ever had to take a Xanax to calm down, you've taken a drug that magnifies the amount of GABA in your brain. GABA inhibits and reduces anxiety. It inhibits neural functioning and reduces anxiety. Glutamate is very important in memory, okay? So you can pay attention and you can learn, but can you remember what it was you were taught? And so your glutamate is going to be very important for memory and learning. And then endorphins are a pain reduction neurotransmitter. So if you've ever jogged and had the runner's high and had a burst of endorphins, you've got the pain reduction that goes along with this internal neurotransmitter. Hopefully that makes sense so far. Again, I will direct your attention to two videos. One explains how synaptic functioning works, and then I was going to show the video, a short video clip today, but I don't dare do that since I had the disaster 20 minutes ago. But I highly suggest you go to the mouse party video that I've got in the course resources folder, and it shows you how psychoactive drugs work, and how, by the same token, how pharmacological drugs work to change the functioning of neurotransmitters in our brain. So, and it's also in the textbook too. Yes, wow, how 1990, Emma. Nobody looks in the book anymore. Okay, right. So, just some general notes about neural communication. I know we are running short on time. Remember from slide number 3, neurons are one-way communicators. So you have afferent and efferent neurons. Afferent are motor neurons, and efferent are sensory neurons. So on Thursday, when we start talking about parts of the brain, what I'm going to tell you is that every node, every nucleus, every module in your brain has afferents, that is messages it's sending out, and efferents, messages it's receiving. And so you can think of your brain as a bunch of different computer modules all communicating together, and each one is sending messages out through an afferent or a motor neuron, and each one is receiving information in through an efferent neuron. I have several bottles of GABA supplements. Do you know? No, I'm afraid I wouldn't be able to comment on that, Hyatt. Or I'm sorry, I forget. Did you say Jennifer was your name? No, I wouldn't know. The PowerPoint. And the PowerPoint is available to copy all this. Absolutely. Okay, so remember that they are one way. Afferents and efferents are motor and sensories. Now, chemicals. Dendrites, you have thousands of dendrites, and they work on a graded response. They're comparing the number of excitatory versus the number of inhibitory, and that can change, and it's a variation, and you have grades of excitatory and inhibitory potentials. But when your neuron decides to send an action potential, it is an all or nothing spark of electricity that runs down the entire length of the axon. So action potentials are all or nothing. Now, neurotransmitters may be excitatory or inhibitory, so they tell the next neuron to send an action potential, excitatory, or they tell it not to send in an action potential, which is inhibitory, and that depends upon the neurotransmitter and where in the brain it is released. But remember, neurotransmitters can have either excitatory or inhibitory effects. All right? And the generation of the action potential does depend upon how much stimulation, excitatory stimulation, the neuron receives compared to the inhibitory stimulation it receives. Okay? So, quick thing that I wanted to mention to you, and I was going to show you the mouse party video in class, but I'm going to send you out to watch that on your own. Psychoactive drugs. What is a psychoactive drug? Well, a psychoactive drug is any drug of abuse that you take, alcohol that causes an altered state, marijuana, cocaine, alcohol, caffeine, legal drugs like that. A psychoactive drug is something your doctor may give you for depression, for anxiety, for schizophrenia. So any molecule that can pass through the blood-brain barrier and is shaped similar to a neurotransmitter can be, if you will, a psychoactive drugs. And psychoactive drugs modulate the effect of the neurotransmitters in our brain. So what they do is they change, the light went off in the lab here. I'm going to have to make it come back on, excuse me. Note to self, figure some way to make sure the lights don't go off during lecture. So your neurotransmitters can be of two types. Some of these neurotransmitters make, some of these psychoactive drugs make our neurotransmitters work better. They magnify the effect of a neurotransmitter. And so we give a general term of agonist to these types of psychoactive drugs. One example might be a selective serotonin reuptake inhibitor, an SSRI, some of you take for depression. And what an SSRI does is it magnifies the amount of serotonin in your brain. On the other hand, some chemicals that you will take work against the functioning of a neurotransmitter. They impair its functioning. And so we'll call these general class of drugs antagonists. And what they do is they minimize the effect of a neurotransmitter. And so one of the first drugs ever invented to treat schizophrenia was Thorazine. And what Thorazine does is it works against the functioning of dopamine in your system, because dopamine, excess dopamine seems to be related to the hallucinations that people who suffer from schizophrenia have. And so Thorazine is what we would call an antagonist. Now, it is way more complex than just antagonists or agonists. These psychoactive drugs work to impair or magnify the effect of neurotransmitters in many different ways. So what I would like for you to do is to go to Mouse Party and watch the short lessons on how each of the psychoactive drugs affects neurotransmitter functioning. And what you're going to see is that there are different ways that drugs act as agonists or antagonists. But the bottom line is they either magnify the effect of a neurotransmitter or they minimize the effect of a neurotransmitter. Any questions so far? Last thing I wanted to mention before we go, and I always give the endocrine system short shrift, and I do apologize. So here's the theory for neurotransmitters. Neurotransmitters work locally. They are released in individual neurons and work at the synaptic site. Hormones are just like neurotransmitters. That is to say, they work on neurons, but instead of being released locally and working locally, they are released globally and work globally. So the endocrine system is a communication system that uses hormones to influence our physiological and mental functioning. That's a mental ability. That's a terrible way to write that. To influence our physiological functioning. And so they're released into the bloodstream, and they work like neurotransmitters with two differences. They're not quite as quick, and they work systemically instead of locally. Now, we're not going to talk about them in too much detail. I'm going to save that for Chapter 10 when we talk about human sexuality. But some examples, or actually we're going to talk about them in Chapter 3. In Chapter 8, emotions, we'll talk about cortisol and testosterone and estrogen. And these are hormones that work on neurons to speed up hearts, to cause brain changes, to cause different changes in our physiological functioning based upon our situation. And we're going to talk about them in more detail, but these are hormones, and hormones function just like neurotransmitters, but they function systematically. Okay? And then you know what? I'm not even going to ask you, well, I might ask you. So your adrenaline glands is where your cortisol is going to be released from, and your ovaries and testes is where testosterone and estrogen are going to be released. And we'll talk about this in chapter 10. Men and women both have testosterone and estrogen in their system. And your pineal, your pituitary, and your thyroid glands are sort of the master control glands that control the release of these different hormones in the system. Okay. Oh, wow, I didn't send ahead to this picture, excuse me. And this is the picture I was talking about without putting it up. And so you'll see your adrenaline glands is where your cortisol is released from, and then the male and the female both have their sex glands where testosterone and estrogen are released, and your pineal, pituitary, and your thyroid glands are the glands that are responsible for controlling the functioning of these glands in your peripheral nervous system. All right, so that's all I had to say today. Do we need to know all the glands on that diagram? No. In fact, Sampata, that's the reason I wasn't even sure I was going to show it. I'm going to save that particular chart for chapter 10 when we talk about sex, and chapter 8 when we talk about the stress response. So that's when I'll probably ask you about those items. So the neuron is the basic unit of a very complex system of communication. Neurons communicate electrochemically, and they are molecules in the body that make neurons either more or less likely to generate action potentials or pulses of electricity. And I suggested to you that psychoactive drugs are drugs that work by modulating the effect of these neurotransmitters. And I did want you to know that hormones are just like neurotransmitters, except they don't work quite as fast, and they work globally instead of locally. All right, so does anybody have any questions? That's all I wanted to share today. Did you enjoy the lecture today? If you would, give me a little feedback here in the chat bar here and let me know what you thought. Is there anything I didn't cover as well as you needed to? All right. Fantastic. Well, hey, don't forget we have optional webinar tonight at 8 p.m. if you want to come. There's optional webinar B tomorrow at 2 p.m. And then, of course, you can come to optional webinar C on Thursday morning at 9 a.m. If you're not going to do the webinar, don't forget to do the discussion board. Well, any other questions? Fantastic. Okay, so before I let you go, yeah, actually, let me go ahead and just let you go. I'm not going to try anything else. I don't want to goof it up. Take care, and I look forward to seeing you in webinar either tonight or in lecture on Thursday. Till then, take care.